Hi, so this is a quick revision to discuss some questions from topic 2, which ranges from medium to high level difficulty. So it's not going to be a long video, but something to start off with if you don't know where to start for your revision. But yeah, let's get to it. So this is according to the 9700 A-level biology according to CAIE syllabus for your reference. All right, so the first question. The diagram shows the structure of a monomer. Okay, so this is actually the structure of alpha glucose. Okay, how do we know? It is by looking at where the position of hydrogen and the OH group is in the carbon 1 of this molecular structure. So here we can see that hydrogen is at the top plane of carbon 1. So this is alpha glucose. Whereas if beta glucose, hydrogen would be here at the bottom, hydroxide would be at the top. And then for those of you who are not sure, how do we number the carbon? Here it would be carbon 1, carbon 2, carbon 3, carbon 4, carbon 5, and carbon 6. Now, I would advise to the students that you should be able to draw the structure of alpha and beta glucose as well. All right, so this is going to be very, very useful for you. In fact, it is stated in your syllabus that you should be able to do so. All right, so I guess the only a tip that I could give you is that for alpha glucose, again, not only that we should remember that the hydrogen is at above the plane, okay, in carbon 1, and then the rest. So probably what you can also take note is here in carbon 3 is where the OH and H is not the same compared to the rest of the positions of the hydrogen and OH in the other carbons. All right, so monomer, a single unit of a particular structure. So in this case, we have amylopectin, sucrose, and glycogen. Now, sucrose is a disaccharide, okay? Okay, it's a disaccharide which contains alpha glucose as well as fructose. Now, fructose has the same carbon number as glucose, so it also has the same formula, which is C6H12O6. All right, and then we have amylopectin. Amylopectin is a structure found in part of glycogen as well as starch as well. Now in amylopectin, okay, the difference between amylose and amylopectin, which are the true structures that we can find in starch as well as glycogen, is that amylopectin has the 1,6 branching of alpha glucose. Okay, they are branches. Okay, and then glycogen is also, amylopectin is also found in glycogen, or part of the structure of glycogen. Again, and of course, just for the sake of revision, just like glycogen, starch and glycogen also have the straight chain of amylose as well, okay, which is from the connection of 1,4 glycosidic bonds. That's why they are straight chains. So if you can remember that, that'll be very, very good. So this one, okay, so these two molecules, amylose, amylopectin, are found in glycogen as well as in starch as well. Right, so glycogen and starch, we know that they have their own purposes. So for starch, basically this is how glucose is stored in plants. And this is how glucose is stored in mammals. Okay, there you go. So to go back to the question, which of these molecules contain this monomer, which is an alpha glucose? Okay, we know that amylopectin does also have alpha glucose, but it's 1,6 branched. Okay, Glycogen also have amylopectin, which contains alpha glucose, as well as the 1,4-glycosidic bonds of amylose molecule. And last but not least, sucrose, which is a disaccharide, which contains alpha glucose, and fructose, which is the isomer of glucose, which is also have the same formula of C6H12O6. So therefore, using this Venn diagram, it is B, because all three of these molecules contains alpha glucose. All right. So the second question, okay, it is basically asking us to find out which of the solution containing the ester bond. So ester bond, that means the biological molecule in our syllabus where we need to know what molecule that contains ester bond is lipids. Okay, that means we need to know what is the positive test for lipids, which is the emulsion test, okay, which is using ethanol. So just a bit of a revision there, ethanol. Okay, you add a little bit of ethanol to the sample, and then you add a little bit of water. Okay, a positive result is when you can see some emulsion or cloudy observation. So for this already, we can start to eliminate the other options because we want to find only the molecules that has lipids, and the one that shows positive result is the solution 1 and solution 3. So therefore, the correct answer is B. Right. For question number 3, they want us to find what is the correct general formula for glycogen. Okay, so just as how I said earlier, glycogen is how mammal would store its glucose. Okay, all right. Now, as glycogen is formed, okay, with the presence of monomer units of alpha glucose, so 1,4-glycosidic bond for amylose and 1,6-glycosidic bond for the amylopectin molecule. Okay, but remember, when these alpha glucose are joined together, they undergo a reaction known as the condensation reaction. Now, with condensation reaction, please remember that water is removed. All right, so I'm going to put there H2O is removed. All right, so going back, right, we know that glucose, alpha glucose, has a formula of C6H12NO6, right? So what we can do is whenever the glucose are joined together, okay, one molecule of H2 is going to be removed, right, for every addition. So we can just minus that with, 2 hydrogen is removed and 1 oxygen is removed. That makes the general formula. Therefore, it is C, C6H10O5N. Okay. Question number 4. Okay, we are given the mean value number of hemoglobin molecule and one red blood cell is 260 millions. What we need to remember is that one hemoglobin molecule, okay, has 2 alpha globin chain and 2 beta globin chain. So now that we have the information that there is a mean value of 260 millions of hemoglobin present in one red blood cell, what you just have to do is you times this value to 2, and you shall get, or you should get the answer C in standard form. All right, moving on. Question number 5, unfortunately, that has been discounted, so we can actually skip. Which general formula, number 6, which general formula is correct for the monosaccharides such as fructose? Now, fructose is basically, again, it is an isomer of glucose, which has C6H12NO6. So, if you simplify that formula, that should give you B. All right. Question number 7. Which formula represents a saturated fatty acid? Saturated means that that fatty acid molecule should only have the C single bonds. right? So if you're not sure how to start, let's just give yourself a scenario, okay, where you give yourself like an example of a molecule of a saturated fatty acid with your chosen number of carbon. So for the sake of this example, to come with or to arrive at my answer, what I did is that I pick a molecule with 5 carbons. So I started drawing like so. Okay, so I started drawing with 5 carbons, right? 2, 3, 4, 5. Now, another thing that I want you to remember is that this is fatty acid. That means it should have a carboxylic acid group or the COO group. So therefore, you can start at that functional group there, C double bond OOH, right? Oh, that was missing. It's a bit unfortunate. All right. And then when you complete that chain, you can start counting what is the formula of this structure. So that's C5H10 and O2. You can try and count them yourself, right? And you can actually notice that from here, with a certain number of carbon, you should have double number of hydrogen and just the same number of oxygen atom for each fatty acid molecule. So with that general formula, you can arrive at A as the answer. All right. And then for the next and also the last MCQ question, this is the trehalose sugar that has a negative result when tested with Benedict's solution. They even tell us that a molecule of trehalose forms 2 alpha glucose molecule when it is hydrolyzed. Okay, so if it's hydrolyzed, it gives alpha glucose, which should give a positive result with the Benedict's test. So we can use the same formula because this is alpha glucose, which has C6H12O6, right? So which row is correct? It should be C because C has 2 alpha glucose molecules, so C12H22O11. Why is that? Because when we have 2 of these molecules together, okay, this is joined by condensation reaction, right? So when 2 alpha glucose molecules join together, it is via condensation reaction. That means water is removed. Okay, so you should minus that total value of, say, it should have been, for example, C12H24O12, right? But then we minus H2O, so minus 2 of hydrogen. That means we have C12H22 minus 1 oxygen. That means O11. So that's how we should arrive with the answer B. And then a sugar that gives the same result with Benedict's as trehalose should be sucrose, because that's an example of non-reducing sugar that we have to know in our syllabus that gives a positive results with hydrolysis. Benedict's test with hydrolysis present. Okay, so that's